Hello, everyone, and welcome to the Society of Systematic Biologists Presidential Address. Today's talk will be given by past SSB President, Dr. Erika on the topic of rediscovering the organism in phylogenetic biology. Before I turn the floor over to Erika, I want to make a couple of announcements associated with the Society, as well as present several awards. To begin, I'd like to let you know that SSB has recently launched a new journal called the Bulletin of the Society of Systematic Biologists, or BSSB. For more information about our new journal, please go to our website, systbio.org, or join us for the business meeting, which will be right after this session at 1:45. If you're an SSB member, you can check your email for information about registering for the business meeting, and I'll also put the information into the chat following the introductory remarks. I also want to let you know about the SSB Symposium that will be held later this week on the topic of diversifying evolution. The symposium will be on Thursday, June 24th from 3 to 6 p.m. Eastern Time, and I've listed the speakers for the symposium there. And I want to give a big thank you to our DEI committee for putting the symposium together. I'm sure it will be a great session, and I hope everyone can attend. Now I have the pleasure of introducing some of our awards given by the Society. The first award that I want to announce is the Publishers Award for Excellence in Systematic Research. This award is sponsored by Oxford University Press, the publisher of Systematic Biology, and is selected by the editor-in-chief of the journal, Dr. Brian Carstens. The award is given to the two best papers published in the journal over the last year, whose first authors are students at the time the research was conducted. I'm pleased to announce this year's award winners: Mariana Braga for the paper Bayesian Inference of Ancestral Host-Parasite Interactions under a Phylogenetic Model of Host Repertoire Evolution, and Nicholas Monjardino Cook for the paper Death is on Our Side, Paleontological Data Drastically Modify Phylogenetic Hypotheses. Congratulations to these two award winners. Next, I want to mention our Graduate Student Research Awards and our Mini Arts Awards. The Graduate Student Research Awards are given for the collection of preliminary data or to enhance dissertation research, for example, by visiting additional field collection sites or museums, and are up to $3,000. The awards are open to students who are members of the Society. In 2020, SSB was delighted to be able to award more than $100,000 to 39 students, and I want to show you quickly the list of all of our award winners. Congratulations to these students. And I want to let everyone know that the 2021 applications will open on August 30th. You can get more information about applying for these awards on our website, again, systbio.org. I also want to let you know that the Mini Arts Awards applications are now open. The Mini Arts Awards are for revisionary taxonomy and systematics and are designed to allow SSB members, so students, postdoc, and faculty, to spend time apprenticed to an expert in a particular taxonomic group or to enhance revisionary taxonomic and systematics research in novel ways. Requests for support for these awards may be in any amount up to $4,000. And again, you can find more information on our website. I also want to mention some of the award symposia that will be happening during this week's meeting. The SSB Ernst Mayr Symposium will be held on Friday, June 25th from 10 a.m. to 1:30 p.m. Eastern Time, and I've listed the students presenting in this award session here. This award is given to the presenter of the outstanding talk in the field of systematics at the annual meeting, and it is open to members of the Society who are students or who have completed their PhD within the last 15 months. This year, because we've had a very unusual year, and last year we did not have a typical annual meeting, we recognize that there should be an opportunity for students who were missed by last year's meeting to also participate in our award competitions. So we're happy to announce that we will have another award symposium, the SSB Excellence Award Symposium, which will be held on Thursday, June 24th from 10 a.m. to 11:30 a.m. Eastern Time. And here are the students that will be presenting in that award session. So congratulations to all these students whose abstracts were picked for presentation in the award session. And I hope that many of you can attend the session. These student talks are always wonderful and really a way to showcase the future of our society. Next up, I want to mention the 2020 IDEA Award winners. The IDEA Award is an award given by three societies, ASN, SSE, and SSB. IDEA stands for Inclusiveness, Diversity, Equity, and Access. The award was created in 2019 and is given to a person or people at any career stage who have strengthened the ecology and evolutionary biology community by promoting inclusiveness and diversity in our fields. The 2020 IDEA Award winners are Dr. Scott Edwards and Dr. Richard Clement, who will present their award plenary talk on Friday the 25th from 1:30 to 2:20 Eastern Time. And the title of their talk is Undergraduate Diversity and Evolution, History, Impact, and Future Potential. And I hope that you can all join us for this award talk. I also want to make sure everyone has seen the announcement of our 2021 IDEA Award winner, Dr. Adriana Briscoe. And we are delighted that she will receive the 2021 award and will present her plenary talk at next year's meeting. Okay, now I have the pleasure of presenting a very special award, the President's Award for Lifetime Achievement. The President's Award for Lifetime Achievement is given to a person who has made outstanding and sustained contributions to the field over the course of many years. The recipient is selected by the three presidents of the society, the president-elect, and the past presidents. And the interval between presentations of this award must be at least 3 years. So the last award was given in 2016. And so last year, in my year as president-elect, Erika Edwards was the president and Cecile Linné was the past president. We realized that it was time to give another President's Award for Lifetime Achievement. And this was one of the very happy situations where, as we were brainstorming, all three of us came to the same person at exactly the same time, which is really a testament to how deserving our winner of the President's Award for Lifetime Achievement is. So I would like to announce our President's Award for Lifetime Achievement for the year 2020 will go to Dr. Chris Simon, who's professor in the Department of Ecology and Evolutionary Biology at the University of Connecticut. In a minute, I'll turn the floor over to Chris to say a few words, but I wanted to give some highlights of Chris's career before doing so. Chris got her BS and MS degrees in zoology from the University of Florida and a PhD in ecology and evolution from SUNY Stony Brook in 1979, studying periodical cicadas, which is what we're probably all very familiar with Chris's work for. After NSF-funded postdoctoral positions at the University of Chicago, Washington University of St. Louis, and the University of Hawaii, Chris took a position as a faculty member as assistant and then associate professor at the University of Hawaii before moving to the University of Connecticut in 1991, where she became full professor in 1996. Her research has addressed so many different things that I'm not sure I can give it justice with a short summary, but I'll try to give it a shot. Chris's research has focused on using molecular systematics and phylogenetic approaches to address questions related to speciation and hybridization, cicada biogeography, especially the spread and maintenance of biological diversity, bacterial and fungal symbionts in cicadas, and the effects of climate change on time to emergence of periodical cicadas. She is widely considered the worldwide expert on cicadas, including both the periodical cicadas of North America and the cicadas of New Zealand and Australia. Her publications since 1994 have attracted more than 13,000 citations, and she's mentored more than 30 undergraduate researchers, 20 graduate students, and 10 postdoctoral scholars in her laboratory. Her extensive work in New Zealand led to her being named an honorary fellow of the Royal Society of New Zealand in 2016. She also has a distinguished record of contributions at the interface of theory and applications, and her work has been influential in guiding methodological development of techniques for molecular phylogenetics. She's also had an early and ongoing role in promoting citizen science and science education, first through the website magicicada.org and now through the Cicada Safari app. And if you haven't downloaded this app, I want to encourage you to check it out. It is extremely cool. And here in Ohio, we're experiencing emergence of cicadas, so it's been especially fun for me to use this app right now. Her CV includes a long list of outreach activities to news and media organizations, such as the Associated Press, BBC Earth, CBS, National Geographic, NPR, and the list goes on and on. Her work on the factors that affect emergence times for periodical cicadas has benefited significantly from these efforts, and it really provides a model for the potential for contributions from citizen science. Chris has also been a tireless advocate and promoter of the activities of SSB, with continuous involvement in society activities from 1993 to today, including services as editor of Systematic Biology and as president-elect, president, and past president. When you talk with Chris, what really shines through is her love of cicadas and love of science more generally, as well as her mentoring of students, postdocs, and junior colleagues. And she's really been a role model for me. So for all of these reasons, I'm delighted to present the President's Award for Lifetime Achievement to Dr. Chris Simon. Chris, if this were in person, this would be the point at which there would be thunderous applause. So please know that we are all applauding and cheering for you from our homes and offices. I could go on and say a lot more, but I'm sure you'd rather hear from Chris than me. So with that, I'll ask Chris to say a few words.
Thanks, Laura, for your very kind introduction, and to you and Erika and Cecile for giving me this award. I was really surprised and honored to receive this recognition that in the past has been limited to methods developers and theoreticians. But of course, no one wins an award totally on their own. And I would like to show a few slides to thank the people who really deserve this award. So let me just share the screen. And then minimize the pictures. So any success that I may have had in my career is in large part due to the support of my lab group and collaborators. I'd like to thank my wonderful Simon Lab postdocs, graduate students, visiting students, and undergraduate research students for their enthusiasm, hard work, and dedication over the years. I'd like to give a special shout out to my first PhD student, Jack Sullivan, and my first postdoc, Francesco Frati, who are seen in the upper left looking quite young. And I'd like to also give a special shout out to my former PhD students, Thomas Buckley, Steve Jordan, and Chris Owen, who are familiar faces in SSB. My collaborators have also been a constant source of inspiration, and I'd especially like to thank 3 of our current project teams. Our deep level cicadidae team was led by Dr. David Marshall and my technician, Kathy Hill, and joined by John Cooley, Chris Owen, Mark Stuckel, and Eric Gordon. With participation by former lab student Chris Owen and other Simon Lab graduate students. Recently, postdoc Eric Gordon and PhD student Mark Stuckel have led the phylogenomics and bioinformatics work in my lab. Current collaborators, in addition to Chris Owen, include Ben Price of the Museum of Natural History, London, Jeff Cole of Los Angeles County Museum, Alan and Emily Levin of Florida State University, Krishnameg Kunti of the National Center for Biological Sciences, Bangalore, Thomas Buckley of Landcare Research, Auckland, Martin Villett of Rhodes University, South Africa, Max Molds of the Australian Museum, Claudio Veloso, University of Chile, Pablo Pisac, Research Center Esquel de la Montaña y Estepa Patagónicas, Pham Hung Thai of the Vietnam National Museum of Nature, and Alma Mohagan of Central Mindanao University. I'm going to go back one slide because I pushed 2 by accident. So I'd really like to thank my Magis Cicada team. It is a collaboration with Japanese scientists led by the remarkable Teiji Sota of Kyoto University and Jin Yoshimura of Shizuoka University. The US side has been a joint venture led by my former postdoc, John Cooley, who's now at UConn Harvard, and a long-term collaborator who's the genius behind Magis Cicada Central. The participation has also really benefited from David Marshall's work and from Gene Kritsky, who is the developer of the Cicada Safari app, not me. I just help promote it. And my field assistant has been Steve Chiswell of the National Institute of Water and Atmospheric Research in New Zealand. In the last 10 years, we've had a lot of fun collaborating with John McCutcheon and his wonderful microbial symbiosis group, and have recently started working with Matt Casson on parasitic cicada fungi. My lab group Symbiont projects are led by postdoc Eric Gordon, who is both an excellent bioinformatician and hemipteran systematist, aided by master's student Master and Honor Student Delaire Haji, who's now at Berkeley, and Jason DeLeonis, with participation from former master's students Russ Meyer and Kate Nazario. Lastly, I'd like to thank the Ecology and Evolutionary Biology Department at UConn and my colleagues at the Society of Systematic Biologists for many years of interesting discussions and friendships. And last, but certainly not least, I'd like to thank the theoreticians and methods developer whose user-friendly programs make our work possible. So thanks very much to all of you. And I'll turn it back over to Laura.
Thank you so much, Chris. And again, congratulations on this well-deserved award. Yes, we're all clapping. Okay, great. I'm gonna share my screen one more time as I introduce Erika Edwards. Erika Edwards is SSB past president and will now give her presidential address. Erika received her BS in Earth Systems from Stanford in 1998, her PhD in Ecology and Evolutionary Biology from Yale in 2005. After appointments as assistant and then associate professor at Brown University, she joined the faculty at Yale University as professor in the Department of Ecology and Evolutionary Biology in 2017. She also serves as director of the Marsh Botanical Garden at Yale and is curator of botany at the Peabody Museum of Natural History. She was recently named a fellow of the Connecticut Academy of Scientists and Engineers and is a winner of the Presidential Award for Early Career Scientists and Engineers, the PCASE Award. She served as an associate editor for the American Journal of Botany, the Systematic Biology, and the International Journal of Plant Sciences. Her research is at the intersection of botany and evolutionary biology with a focus on understanding the evolution of diversity in plants. Her work is focused specifically on evolution of C4 and CAM photosynthesis, on leaf form and function in viburnum, among other things in viburnum, and a range of evolutionary questions in a group that includes the cacti. So with that, I'm very happy to turn the floor over to Erika for her presidential address. Thanks, Erika.
Thanks, Laura. And congrats again, Chris. We're so happy that we could finally honor you this year, even though you officially got the award last year. So thanks for joining us. Let me see if I can do this. Let's see. Okay. Are my slides good? Okay. Well, so thanks, Laura, for that really kind introduction. I'm going to just sort of jump right in because we're already about, I don't know, quarter of the way through our slot here. And before I get into talking about science and systematics, I want to just give a shout out to the leadership team that I had the pleasure of working with last year. So I am the past president, meaning that I was president during 2020, which we all know was a year like no other. And it was full of sort of new challenges and really kind of stretched us all in many, many dimensions. So it's a very challenging year to get anything done. And I just want to sort of acknowledge the team of people that were keeping SSB afloat and alive and actually engaged and active during this difficult year. So Laura and Cecile is my sort of presidential team. We worked really well together. I also want to give a shout out to Kelly Zamudio, who was our joint meeting council rep, and she actually stayed on for an additional year. Her term was up and she's like, oh, I'll just do 2020 for you. And I don't think she was ready for that. It was a very interesting year to be the joint meeting council representative. And while I'm here, I also just want to sort of acknowledge Tracy Heath, who I don't think a lot of people understand this, but she has actually served SSB in one form or another for 9 consecutive years, first as a council member, then as an awards director, and her last term here is as communications director. I would say she is the institutional memory of the society. And I just want to congratulate Tracy and thank her from the bottom of my heart for her dedication to SSB. I don't know where we would be without her. Also, 2020 was a crazy year for evolution conference activity. So one thing that's really fun about this conference, of course, is that it's this tri-society endeavor. And so all the evolution societies get to interact with each other. And I would say, even though I didn't get to meet any of these three wonderful people, Susan, Ruth, and Andrea, in person, we interacted a lot more than we probably typically would, because we had a lot of figuring out to do in terms of how are we going to cancel the 2020 meetings without putting all three societies in bankruptcy. It was stressful, but sort of inspirational time. I really enjoyed working with these three people, and they're some of my favorite people. And I hope one day we can actually get together for a drink after a talk someday and meet in person and reminisce about that crazy year. And while I'm here, I also just want to sort of acknowledge these two guys, Mitch and Howard. I think I didn't really realize what went into making these meetings happen until 2020, when we were trying to figure out how to actually cancel one. I realized the amount of work that Mitch and Howard do as co-chairs of this conference. And because we all come to these conferences all the time, I don't know that these two really get the recognition that they deserve in terms of the amount of work that goes into making sure this conference is a success. So I just want everyone out there to just give a little applause to Mitch and Howard and acknowledge how grateful we are for all the work that they do behind the scenes. All right. So when I was elected president of SSB, I of course thought it was a great honor, and it was, and I've really enjoyed serving. But I have to say, my first thought that kind of wandered into my mind was, yeah, but am I a systematist though? What am I doing here? And I'm wondering how many people in my generation have a similar relationship to the word systematist or systematic biology. I never really identified with being a systematist. And so when I go back through time and I think about my training and where I was as a graduate student and how I came to be where I am right now, I think back to what I thought a systematist was, when I was 25 or 26 and deciding to actually try this career out. And I think, in my view, I think I thought a systematist was someone who could, with confidence and accuracy, apply all of these various 40 vocab words to describe the margin of a leaf, right? So a systematist would know if a leaf was dentate or cerulate, whereas I would call a leaf toothy or not. And while I was just kind of blown away, and I have mad respect for this level of skill and sort of observation that I attributed to systematists, I knew that was never going to be me. And so I've been thinking about this a little bit more and trying to figure out if I was way off during that time or what people in generally think systematic biology is. And so I went, when I was thinking about this talk, I went back to sort of the very first, I thought, well, why don't I just go back to the beginning of the society in terms of what was our very first, what were we talking about in the initial volumes of our journal, our society journal, which was called Systematic Zoology at the time. And so this is our very first volume, our very first issue. And what's really funny to me is that, right there on the cover, in the very first volume of our journal, we published a 3-part essay describing what Blackwelder and Boyden thought the nature of systematics was. And it was a real exploration into what the term means. So in some way, I think we've always been asking ourselves this question and wondering what it is that we're actually doing, what makes us systematists. And that essay didn't really move me one way or another, but what it did do is actually point me to this other quote that I found really interesting. And so this was something that Simpson wrote in 1945 when he was trying to explain, his view about taxonomy. And in this case, you can substitute the word taxonomy for systematics, I think. And I like this explanation of it. So he says taxonomy or systematics is at the same time the most elementary and most inclusive part of zoology. Most elementary because animals cannot be discussed or treated in a scientific way until some taxonomy has been achieved. And so that's kind of, I think, my old-fashioned view of what I associated systematics with. But this second part is really different and most inclusive because taxonomy in its various guises and branches eventually gathers together, utilizes, summarizes, and implements everything that is known about animals, whether morphological, physiological, psychological, or ecological. Now, I never came across that quote. Oh, and I kind of fixed it for him, right? So I'm a botanist, and this makes more sense. I never came across this quote as a graduate student, and I kind of wish I would, because it might have opened my mind up to thinking more broadly about what systematics can be. And instead, I would say I came of age, if you will, scientific age, during the real heyday of what I would call phylogenetic biology. And I think phylogenetic biology in one level, at least initially, was maybe a rebranding, if you will, of systematic biology, but it's also a little bit different, right? And so I came to this field as an ecologist. So I started off loving plant ecology. I started getting interested in evolution a little bit, but what really sunk me in was the way that people were using phylogenies at that time to ask very pointedly ecological questions. And so I think this period of time in the 90s and early 2000s was a period of sort of really branching out for systematic biology, where we were really taking phylogenies and just throwing them at every different kind of biological question you could come up with to see how we could be useful to other fields. And the thought of phylogenies as a tool is something that I grew up thinking, and that was kind of my worldview at that time. And so, my thesis work, I'd say, is a prime example of like classical phylogenetic biology. In other words, I started working on this lineage, the Portulacini, this wonderful clade that we'll talk about later. And I became interested in this group, not because of the group itself, but because I was interested in a very specific question. And I thought that this group and a phylogenetic approach to answering that question is just what I needed. So this is from the very end of my PhD, doing fieldwork in Bolivia. And you'll see me standing in front of a big cactus. So the Cactaceae is sort of the sort of enigmatic lineage of the Portulacini. And what I was really interested in was how the cactus life form evolved, because it's a very weird looking plant, right? So I was really interested in the changes in form and function that would be required to go from, this little plant on the left here to something like this. And so I did what a good phylogenetic biologist would do. I actually sort of backed out from the cacti. I tried to change my, get my taxon sampling whereby I could bracket where in the phylogeny the changes occurred so I could understand the order of events. And that was really fun. And so that was kind of the start of it. What's interesting is that I've never been able to let the Portulacini go. So they kind of, even though I answered the question that I started with, I keep coming back to this clade. And we'll get back to that later in the talk. Now, I would say over the last 20 years or so, so about when I finished my PhD to the current time, phylogenetic biology or systematic biology, or we'll figure out what we want to call it later maybe, has actually undergone another whole round of revolution. And so I think, two things sort of started happening. One, the shift over to molecular phylogenetics, so no longer inferring phylogenetic relationships based on morphological data sets, but just doing a lot of sequencing and using molecular data sets to do the work. That released a huge sort of data revolution. So now we have so much more data than we could ever imagine for our use in phylogenetic inference. And this makes both data matrices that are deep and wide. So we can use thousands of characters and we can use thousands of tips. And what's happening is tree building of a sort of unprecedented scale. And at the same time, we've had, I would say, decades of a decades-long frenzy of methods development. And some of my, presidential team, Cecile and Laura, are at the forefront of that work. We really are sitting in a situation now where we have such a wonderful array of sophisticated methods, both for phylogenetic inference and also just for character evolution, diversification, a whole suite of comparative methodology questions that we can ask. And this has been amazing. So I would say, the field of systematic biology is bright and healthy and vibrant right now. But at the same time, there's trade-offs to everything in life. And so if we're investing a lot of our intellectual capacities in, increasing our data availability and the maturity at which we actually can analyze that data, and the sophistication, we are losing something. And I'd argue that one thing that we've lost a little bit is the careful attention to observation of the organism. So if you go back to old-fashioned systematics, right, where you actually had to derive a morphological data set in order to, build your character matrix to infer the phylogenetic relationships of your organisms, you had to spend a lot of time looking at your organisms. You had to spend months and months and years, and you actually had to very carefully understand how they're put together, because, the responsibility of the systematist was to identify, to correctly atomize the organism into comparable parts. And those parts had to be homologous across organisms. And to do that accurately, you had to really understand how an organism was put together. And you had to know everything about it, and what was variable and what wasn't, and what was important and what wasn't. And that kind of sort of attention to detail is something that we don't need to do anymore in order to build a tree, or in order to do some sort of trait analysis on our tree. We don't need to do this. So the question is, should we let it go? And I'd argue that we should not let it go, and that actually systematic biology is kind of where it's at in terms of understanding the organism. And so this attention to the phenotype is something that's in the back of my head. And another thing that I think we might be losing, and if we completely move on to the 2000 to 2020 version of systematic biology, is that actually continued study of a particular group of organisms kind of gives you a perspective and a creativity and an understanding that you wouldn't get just by jumping around from group to group. So while I've been thinking about these things, I've had the huge privilege of actually initiating this, 14-year collaboration with my former PhD advisor, Michael Donahue. So we've been working together on viburnum, questions in viburnum, which is another clade that I'm growing to love deeply. And what's been really fun about this period of time is that Michael studied viburnum for his PhD back in 1980-ish, and maintained an interest in it, but then went on to do a whole bunch of other different things with his life, right? And so then just more recently, over the last decade or so, as he's actually planning to retire in the next couple of years, he's come back to viburnum. And it's been really amazing to watch the sort of long-term nature of a relationship with a clade. And both the immense sort of insight that you get from that and the immense satisfaction that you get from it. And so when I look forward, this has been very inspirational to me. When I look forward in my life, what do I want when I'm getting ready to retire? I want to have the deep knowledge of a clade that I'm watching develop in Michael. And it's only because he's been doing this for 40 years. And in this collaboration, we've also been sort of chatting at length about what this perspective is, or what we're trying to say, and what does it mean, the kind of integrative work at a clade level that we're doing, what is it? And we've been coming up with names and things, and we're going to write a little paper about it. But what struck me about this quote is that it's more or less kind of what Simpson highlighted in 1945 as his systematics. And the part that I really resonate with in this quote is the idea that your job as a systematist is to gather together everything that is known. And that everything is infinite, right? And it's when you gather together everything that is known that you start making connections between things in new and creative ways. And so Michael and I have been calling this approach the model clade approach. And maybe that's right. Maybe it's time for a rebranding yet again, which is what we tend to do over time. Alternatively, maybe it's just a way to like resurrect the classical or, the most inclusive kind of systematics. And to think about systematics really as a way of knowing, as a way of being, as a way of approaching your questions in science. And in terms of focusing back in on the organism, I'd argue that now is the best time in the world to do that, because today, modern day systematists like us, we have superpowers. In other words, we are not relegated to looking at our organisms as, dried branches on an herbarium sheet. We have an infinite number of ways to look at our organisms. I would say now's never been a better time. And that if you're interested in the functional genomics of your system, you don't need to work on a model organism. You can work on whatever you want. In terms of visualizing just the inside and the outside of the organism, we have amazing powers of anatomical visualization that we never had before. If you want to look across thousands of species, the things that we can do with rapid phenotyping now is just also kind of amazing. So it's like the opportunities are endless. And now is the best time to dive back in and rediscover the organisms in phylogenetic biology or in systematics or whatever we want to call ourselves. So with that, I'm just going to jump in and give you kind of one example of the way that we've started looking at our organisms that I'm finding super exciting. And this work is taking place back in the Portulacini. So the Portulacini is this clade that we are sort of wedded to at this point in the lab. We're very fascinated with them. It's about 2,200 species, most of which are cacti. So the cacti are kind of the big group in this clade, but there's a lot of other really interesting things going on here. And a lot of people over the course of my time as a faculty member have been like sort of roped into this, and I hope they continue on and be Portulacini heads for the rest of their lives also. And so the one little clade within the Portulacini I want to talk about today is Portulaca, also known as purslane. This is a sort of unassuming little plant. It's a worldwide weed, so you've definitely stepped on a purslane. You've probably eaten one, or you could eat one. They're actually really tasty, and they're actually crops in some regions of the world. And it turns out that they are also a photosynthetic marvel. And in order for you to understand why they're fabulous, I have to give you a mini lecture on plant photosynthesis, just to get everyone up to speed. As Laura mentioned, we do study the evolution of different photosynthetic metabolisms in our group. So very briefly, most plants use what we call C3 photosynthesis. However, there are these alternative metabolisms called C4 and CAM. And I'm going to tell you a little bit about how photosynthesis works, so you can understand why these types have evolved. So I would argue that the ultimate sort of trade-off that's governing so much about how plants function and all of their sort of anatomical and physiological adaptations to different environments revolves around this one trade-off, and that plants have these little pores on their leaf surfaces called stomata, and they open and close. And what we know is that plants make their living by actually fixing atmospheric CO2 and turning it into sugars, right? So they're the primary producers of our planet. And the way they get the CO2 is that when they open these little pores called stomata, the CO2 diffuses in passively just along a concentration gradient. The trade-off for plants is that when they have the stomata open to do this, water diffuses out along its own concentration gradient. So every little bit of CO2 a plant needs to make a sugar comes at a cost of a loss of water. And this trade-off is something that plants balance very intricately all the time. They're just opening and closing these pores constantly to sort of regulate this balance. The important thing to know from this is that because CO2 is passively diffusing in, what it means is that the CO2 concentrations inside the leaf must always be lower than those in the atmosphere. So the atmospheric CO2 concentrations set this ultimate ceiling on what the CO2 concentrations inside the leaf can be. And that can become a problem for photosynthesis, because the Calvin cycle in Rubisco and the main process by which sugars are made works really well when CO2 concentration levels are relatively high and temperatures are low. And when those conditions are not met, plants actually are presented with the real problem, and that rubisco actually ends up interacting with oxygen more than it does with CO2, creating a whole bunch of metabolites that the plant has to process that are energetically expensive, and engages in this process called photorespiration, which actually produces CO2 instead of fixing it. So this is a real problem, and plants struggle when CO2 concentrations inside their leaves get too low. And so what C4 and CAM are is really just an evolutionary solution to that problem. You can think of them as a CO2 pump or a carbon concentrating mechanism. And what they've done is really just go into the plant biochemistry library and borrow a few biochemical modules that all plants already do. So it's a repurposing of things that a plant already has. And they use these enzymes to create a elevated CO2 concentration around the Calvin cycle. So the important one here is PEP carboxylase. So basically this enzyme is first interacting with CO2, turning it into a 4-carbon acid. And then that acid eventually gets re-released, decarboxylated, and CO2 is re-released around wherever the Calvin cycle is. So C4 and CAM use the same biochemical module. They just implement it a little differently, and I'll tell you about that. Now, what we know about the origins of these metabolisms is that they're relatively recent in time. In other words, we think that global atmospheric CO2 concentrations are the sort of ultimate global driver of the evolutionary origin of these CCMs. And we think most of them have occurred within the last 30 million years. And in reality, really like the last 10 or so million years. So they're a very recent sort of innovation in land plant history. And what's really cool about them and why we like to study them is that they do take this same biochemical module, but they implement the separation of that PEP carboxylase and rubisco differently. So in a C4 plant, you've got basically a 2-cell photosynthetic system where CO2 goes into what we call these mesophyll cells, these sort of general leaf tissue cells that can photosynthesize. It's fixed there first as a C4 acid, and then it's moved into these very specialized group of cells. This is a cross-section of a leaf called the bundle sheath. And these cells are where the Calvin cycle operates. And this is where the CO2 is re-released. And rubisco is tricked into thinking it's living in this high CO2 world. So the key anatomical trait that C4 plants need in order for this to work is a high bundle sheath to mesophyll ratio. So they have a very specific kind of anatomy that's required for C4 plants to work efficiently. What CAM plants have done instead is take that same PEP carboxylase module and actually separate PET-C and rubisco temporally. So in a CAM system, everything's operating in the same cell. It's just that stomata open at night and are diffused in and are fixed. CO2 is fixed by PET-C during the night, where its C4 acid is actually stored in the vacuole, which is kind of like this storage organ for plant cells. And then during the day, the stomata close, the C4 acid diffuses out, is decarboxylated. And so the anatomical traits that this requires are basically succulents, right? And so we see the correlation of strong CAM photosynthesis and succulents all across the tree of life. So you need very large cells with very large vacuoles. And you also need sort of low intercellular air spaces. You need those cells to be all jammed up next to each other so you don't actually leak out a lot of CO2 during the day. Okay, so same biochemistry, different sort of implementation. And with regard to CAM, I just want to make the note that actually there's a really interesting and broad range of phenotypes that CAM photosynthesis describes. So if you have like a typical C3 plant over on this side and something like a cactus or an agave, what we call a strong CAM plant that has really committed itself to CAM being its primary metabolism, in between these things, you have a lot of other really interesting plants that do a little bit of both. So there's a great diversity of plants that we call C3 plus CAM, where they're actually, when things are good and they're not stressed, they're C3 plants. And when they get a little bit stressed out, they turn on a little CAM cycle. And lest you think that these C3 plus CAM plants aren't important, I just like showing this photograph. We were doing some collecting of Systanthe, which is a really cool group in the Portuguesini. This was out in the Atacama Desert during one of their amazing blooms. And you're looking at a monospecific stand of Systanthe longoscapa, which is one of our Montiaceae. that is a C3 plus CAM plant. So these can be very ecologically dominant species in certain environments. Now, when you look across the tree of life, the plant tree of life, you can see that although this is messing with the primary metabolism, and it appears to be kind of complex, and that requires a lot of biochemistry, gene regulation, and anatomical specialization, these syndromes have evolved tons of times. And this is obviously still an underestimate. So we're really still trying to understand exactly how many times C4 and CAM have evolved. But they're certainly, if you add them up together, it's over the hundreds. And what's interesting is that they have these sort of slightly different distributions, and that I would argue that in terms of the clustering, so we know more about C4, we think it's evolved maybe 70 times independently, but fully 2/3 of the origins of C4 are just in two clades, one in grasses and the other in Amaranthaceae. Whereas CAM itself appears to be slightly more diffuse. In other words, it's maybe an adaptation that's more accessible to a greater variety of organisms. And what's important for this talk is that we tend to think, even though they share this biochemical module and they sort of seem to be ecologically co-occurring in lots of places, we think their evolutionary trajectories are largely separate from one another. In other words, there's a whole bunch of reasons why we think that certain steps along the evolutionary trajectories of C4 and CAM are almost antagonistic to each other. So it would be very difficult for one plant to evolve C4 and CAM at the same time. And a lot of the work that we've done on this problem has been in the Portulacini. So the Portulacini happens to be a really good clade for understanding C4 and CAM evolution. And maybe, I don't know if it's what came first, the chicken or the egg. Am I studying C4 and CAM because I love the Portulacini, or am I studying the Portulacini because I love C4 and CAM? I don't know. I think it's maybe both at this point. Anyway, it's a really great group. So we've got multiple origins of different kinds of CAM phenotypes. And we also have, interestingly, multiple origins of C4. And the one that I want to talk about today is this guy right here, Portulaca, right? And so what I didn't tell you about why Portulaca is a marvel is that it is a C4 plus CAM plant. So just as I was telling you that we think there's good reason to believe that these two metabolisms would be incompatible. They're not, because they coexist in this plant. And what's really interesting to me is that plant physiologists have known this since 1980. So it's not like this is something that we discovered at all. This was a study by Cacti and Kennedy in 1980 demonstrating that we knew that Portulaca was a C4 plant, and if you drought it or change its day length or stress it in a variety of ways, it will turn on a CAM cycle and start accumulating acids overnight. And what's kind of fun is that, the relevance of this, I think, maybe became more apparent to us as systematic biologists, because we've been gathering together, everything that is known about the Portulacini. And when you put this kind of paper in context, in evolutionary context of the other kinds of things that you're studying in a clade, its importance changes, and you realize we've got to figure out how this is. And so that's what we're doing. And so we're really focusing on Portulaca right now. And they're a wonderful group of plants. There's about 100 species. They're really diverse, and there's lots of other cool questions to ask about them in terms of biogeography, life history, evolution, polyploidy, cleistogamy. There's a lot of fun questions that will keep us busy in Portulaca for a while. And I just want to give a shout out to the other folks in the lab who are here at this conference here. But please, if you want to hear more about Portulaca, Nora Heafy just gave a wonderful talk this morning in the macroevolution section about the ecology, the niche evolution when you evolve C4 on top of a CAM system, what happens. And then my grad student, Ian Gilman, and Jose Moreno Villena, who's a postdoc in the lab, they have a double header in transcriptomics tomorrow, where they're going to go into more detail about the little bit of stuff I'm going to talk to you about today. And so what do we know so far about how these strange organisms evolved? Well, we have a lot of data that sort of point to the idea that ancestrally, Portulaca was what we call the C3 plus CAM plant. It was a C3 plant that could facultatively engage in CAM under different conditions. And what's really interesting is that it didn't evolve C4 once on top of that. We think it evolved C4 maybe 3 separate times, sort of in parallel on top of this ancestral C3 plus CAM state. And what we're really interested in knowing now is actually, and this is a little funny for us because we're sort of going out into new territory, we want to understand mechanistically how it's working. And we think that understanding that will help us to understand how it evolved. And so to get right down into looking at the organisms now, what I want to do is show you sort of a little cartoon of what we mean by mechanistically, how does it work? Specifically, we're interested in the spatial integration or segregation, we don't really know, of these two metabolisms. How intertwined are they within the leaf itself as an organ? And so if you do take this little cartoon of a cross section of a leaf, this is a generalized version of what a portulaca leaf looks like. There's actually a lot of variation, but let's just go here for now. So in this case, we have this outer ring of vein tissue and photosynthetic tissue. So the yellow is the photosynthetic mesophyll. The red here are those bundle sheath areas where in a C4 plant, that's where rubisco is and the Calvin cycle. And then a lot of portulacas have this sort of specialized water tissue zone where they store a lot of their water. That's where their succulence is. And so how are C4 and CAM connected here? So one hypothesis in the literature is that, well, because they're using the same biochemical modules and all of these genes have to be regulated at different times and in different places, they can't possibly be coordinated. So one hypothesis is that they actually are working in totally independent tissue areas of the leaf. So you've got CAM operating only in the water storage tissue, and then this is sort of your C4 zone. A second hypothesis from the literature was that actually, why does it have to be that way? In other words, maybe you could have a 2-cell CAM system whereby you've got CAM turning on in the mesophyll at night, making its C4 acid, its malic acid that's stored in the vacuole. And then during the day, the malate just kind of diffuses back out and gets taken up into the C4 cycle. That seems like a really good idea because they're using the same metabolites. So why can't the C4 system just metabolize the malate from the CAM cycle? And then the third alternative is actually that you can't make a 2-cell CAM system. That's too difficult. It's never been seen before. And CAM is still operating as a single cell cycle and just happens to be operating only in the bundle sheath cells, which is where the Calvin cycle is, which is where it needs to go. So those are the hypotheses that we kind of set out. And I need to sort of highlight Jose here. So Jose Moreno Villena has been a postdoc who's been working on this for like 4 years. Super brave and super skilled. So what he did was, we ventured into a place we had never been before, which was how do we analyze gene expression spatially within a single leaf. And so he took 2 approaches. The first was to use laser capture microdissection on frozen materials. And what you do in this case is that you actually, perform all your experiments, get your morning, night, droughted, not droughted tissues. So it's a lot of tissues to even show how this would work. You flash freeze them to preserve the RNA. And then you actually put them on a slide and kind of cut out the tissues so that you can actually sort of isolate, say, bundle sheath tissue here from mesophyll tissue here, from water storage tissue over there. And then you do that over and over. And so Jose was originally going to make really good comparative biologists out of us and do this across like 8 species and 3 origins. And that was not realistic. So instead, I'm going to tell you, we decided to focus on 1 species, Portulaca oleracea, and see how far we can get. And so this is kind of just a zoom up of the kind of fine-tuned sort of dissecting that he had to do with a laser to do this. So he did that. And when you look at actually all of his samples, what you see is that his laser dissection worked really well. So he has a good eye and a good hand, and he was actually really able to repeatedly and accurately separate out the bundle sheath from the mesophyll tissue. Now, at the same time that he was doing this, we came across this new technology and we thought we had to maybe try it. So in terms of how you want to view your organism, to me, this is the ultimate, in that there are technologies now available to us where you can really just kind of order a slide, take a section of tissue that you are interested in, plop it on your slide, do a set of reactions, sequence what comes out the other side, put it back on the original slide that you laid it on in terms of your anatomy, and actually sort of achieve an in situ perfect photograph of gene expression. In other words, across the whole leaf tissue. And I'll go here to see if this is a paradermal section of a whole portulaca leaf. And you can see here the veins, and we can identify where the mesophyll and the bundle sheath are here. And this is a big little part of water storage tissue in the center of the leaf. You can actually layer that back on to these data, these transcripts that you get from this kind of slide, and actually just kind of able to see gene by gene as you go through where they are located in your leaf. So it is actually a photograph, and it blows my mind in terms of what you're able to do with a non-model system. So we don't even have a genome of the species, and yet we were able to get a photograph of gene expression across a leaf. And this is kind of what it looks like. So here we're just kind of going through, I love it, it's like a light bright experiment. It sends me back to my childhood, where you can actually take any gene of interest that you want. These are all our favorite photosynthesis genes, and just like light up their degree of expression across the leaf. And the one thing I want to emphasize here is this big black hole in the middle. This is the water storage tissue. And so right away, we already have good evidence that there's no like separate CAM system operating in the water storage tissue. In fact, there's nothing really going on in the water storage tissue at all. It's a wasteland. It's basically a water reservoir, and very low gene activity. But when you sort of put this all together, what we're finding is that we're able to actually very accurately identify mesophyll markers and bundle sheath markers using a set of genes and using the anatomy. And in general, the Visium, this sort of photograph approach, lines up quite well with the LCM approach, and that both of our approaches to spatial gene expression are telling us the same story. And the story is that really Oleracea, portulaca oleracea, is operating a 2-cell CAM system. So this is something that hasn't really been described before. And what we've learned is that C4 and CAM are operating in the same exact mesophyll cells, C4 during the day, CAM during night. And the malate that is being formed by the CAM system is simply being shunted directly in to the C4 cycle. And it's beautifully simple if you step back and think about it, like, well, how else could it be? That's a perfect way for it to work. What's interesting is that it also opens up the door to, a bunch of interesting questions in terms of crop improvements and ways in which we might be able to put a CAM cycle into something like corn to make it just so much more drought tolerant. It's a much simpler system than we thought it would be when we started this project. And so I just want to end by saying, this is a very strange way to sort of look at my organism as a systematic biologist. And when I step back and I look at the trajectory of what our lab has done and what we're doing now, could I ever have predicted where we are? And the question is, the answer is clearly no. I never, ever thought that I would be this interested in biochemistry. And that's what's sort of beautiful about this approach, this model clade or systematics as a way of knowing approach, and that over the years, we have basically continued to, love and investigate these organisms, and we let the organisms tell us where the questions are. So if you kind of let yourself be guided by your clade, what ends up happening is that you become a very interdisciplinary scientist, and it keeps your science fresh and creative. It keeps you learning. I've had the most wonderful time with Ian and Jose and Hauran, the three people who are working on this project. We have these weekly meetings where they teach me about biochemistry, way more biochemistry than I ever thought I would ever want to learn. And I love it, and I'm eating it up. And that is one of the true benefits, I think, of being a model clade person. And I want to end by this, just one other sort of sentiment or thought that I've had over the years that I think is an unfortunate reputation of systematics that I think we can change. And I think it's not even true. And that another sort of feeling that I think that people get from systematists is that it is almost like a very sort of territorial field, and that, you know, people have their group of organisms and there has to be one expert, one world expert on a particular group of organisms. And I find that to be absolutely untrue. And it's something that I would like to change about our reputation. In other words, the Portulicini is a wonderful clade, and I'll be studying it for the rest of my life. And I really hope other people are too. And that if you're on a mission to know everything about an organism or a group of organisms, then there's room for everyone. I mean, it's endless and infinite, the things that there are to do with a particular group. And so I'm hopeful that we can sort of shed that old reputation as a little dusty and think about, you know, systematic biology, model clades as places where there's an infinite number of possibilities for people to be involved and to study different elements of them. And so with that, I think I'm going to end. I think I'm actually on time. I can't believe it. But I want to thank everyone for listening. And I, of course, need to thank all the members of my lab, past and present. This picture is actually not up to date. I'm so sorry. I think I stopped adding people a couple of years ago, but I have some more work to do there. But I've been, you know, really just so lucky to have just really wonderful people join our group, and it's been a lot of fun, and I'm always looking for more, so, you know, give me a ring. And with that, I think I can take questions. I am going to maybe stop sharing so I can see. Is that okay? Okay, so I can actually see some of the chat. Okay.
Yeah, great. Thank you so much, Erika. I really like how you presented the perspective of knowing the whole organism and then gave us a great example of how to do that. And I hope you have a chance to skim through the chat, because there was toward the end of the talk there was just a string of comments that used words like amazing, cool, neat, incredible, nuts, and just, you know, over and over amazing. So I didn't get a chance to count them all because I was busy watching for questions, but clearly lots of enthusiasm for this talk and for your approach. So thanks very much. We do have several questions in the chat, so I'm going to convey those to you, or sorry, in the Q&A, okay. I'm going to convey those to you. And I'm going to start with actually the one that came in most recently, because it's related to the end of your talk. And that question was, how do you account for the damage of cutting the leaves and the metabolic changes it produces in the spatial gene expression analysis, or are the leaves still attached to the plant?
Yeah, they are not still attached to the plant, but what we do is, like, as soon as we cut the leaf, we throw it in liquid nitrogen. So it's immediately like flash frozen. And I think that's as good as you can do with this type of stuff. So the idea is that you immediately freeze it and keep, you know, kind of just suspends the RNA as it is.
Okay, great. I'm going to also read one that just came in, since I think it could be related, although I haven't read the whole thing yet, so I'll do my best. So does a two-cell system mean there are multiple paired systems, or does it mean that there are two types of cells and some interaction in between? And how do you distinguish between these two with your data?
Yeah, so yeah, there's a lot going on there. And I encourage you to go to Jose's talk tomorrow at Wednesday, where there'll be more detail. What I mean by a two-cell system is simply that you have that CAM is actually making the C4 acid malate in the mesophyll cells, so a population of cells, a cell type. And then it is actually, in the next day, that malate is being moved over into a different population of cells, the bundle sheath cells, to be decarboxylated. And so that's what we're calling a two-cell system, which is distinct from most CAM species, where we think the whole cycle occurs in just one individual cell. So it's the movement of a metabolite between cells that we're emphasizing in that phrase. Does that make sense? We think there's also some CAM going on in the bundle sheath also. So in reality, we think portulaca is a mix of a 2-cell CAM and a traditional CAM, but that most of the malate's being made in the mesophyll.
Okay, great. A couple more questions still related to this, so I'm going to continue with this sort of thread. So one is, do you think that other species of portulaca with C4 plus CAM also use the 2-cell CAM system, or maybe something else evolved there? Since it was what we took away is that it evolved a couple of times. So is that something that you're going to check?
Yes, that's the next plan. Now that we had this working in Oleracea, we're going to try to make sure we do the same set of analyses in all the different C4 origins. And a talk that is related to this is not spatial resolution, but Ian Gilman tomorrow is also going to talk about comparing gene expression networks across 2 different origins of C4. So amylase, which is one origin, and then Oleracea, which is a separate origin. And he's already finding some significant differences that make us think that we will find differences in how these systems are integrated across the different origins. Yeah.
Great. And then does the jack of all trades, master of none principle apply? So are organisms that do both of these less good at either of them?
Yeah, we actually think so, to be honest. Yeah, I mean, portulaca is a wonderful C4 plant, but there's things about it that are a little odd. In other words, when you look at the anatomy of portulaca, it is not, I would say, optimized. So when we think about the greatest C4 plants in the world, they're mostly in the grasses, right? And they have really, really high photosynthetic rates, and they're just amazing bomber plants. And they are the ones that have these super big bundle sheath cells. So they've really kind of gone in for this. Portulaca is like a little bit succulent. Its bundle sheath cells are a little smaller than maybe they could be. And one thought is that its anatomy isn't optimized for either of these pathways because it's actually trying to use both of them. So yeah, great comment.
Great. Yeah, so here's a broader question, not so much about the specific organisms, but about the approach. And I really like this, and it's gotten a bunch of upvotes, so we'll be interested to hear your thoughts. So I'm going to read the whole question. I love your approach so much of being driven by knowing more about the organism. This has been my dream as a scale insect biologist. I noticed, however, that as an early career researcher, it is challenging to find a department that understands the model clade approach, especially if the clade is not, in quotes, popular. Do you have any thoughts on this?
Oh, just stick to your guns, I don't know. I think when you're just getting going on a clade and nobody thinks it's all that interesting, that might be a time where you do want to sort of envelop it in questions that people actually do find interesting. So, one way to make just any old random clade seem relevant to somebody is to say, but you know what, it's really good for asking this particular question, and this question is really general, and so this is what I'm going to do with it, or something like that. And then eventually, of course, 10, 20 years later, you'll be flooded with questions because you'll know so much about the group. But I feel you, like that's tough, actually. And I'd be happy to talk about it offline too, if you want. But my first thought is just cover yourself, and like just make sure everyone understands that you're still really interested in general problems, and that you can be interested in general questions and be interested in a very specific clade at the same time. They're not exclusionary.
That's great. And a follow-up question to that was that a model clade approach often requires a willingness to learn difficult new techniques, which may be expensive and take time to acquire. So do you have advice for early career researchers on when or how to make that leap?
Yes, I think that's true. And admittedly, the stuff that we're doing with the spatial gene sequencing right now is not cheap, right? So there is a time and a place for when you have the resources available to do certain things and not others. I think on one end, I think accessibility to that kind of stuff is going to just keep increasing because sequencing continues to decline in costs, and things just end up getting cheaper over time. At the same time, there's plenty of things to do that don't take a lot of money. And, going back to, it's not, I didn't want to denigrate the dried branches on a sheet, because you actually can get a lot of information, really interesting and relevant information from that kind of data also. And so I think working with what you've got, and also, finding collaborations sometimes is a really good way to sort of expand your resource availability, finding other groups that you can work with and collaborate with.
Great, thank you. We have a question. Do you see any paths to further non-obligate metabolic systems, or what triggers exist that would cause a reversion to earlier metabolic systems?
So in terms of C4 origins, we do not have any strong evidence of a reversal. So we think in some ways the C4 trajectory is almost unidirectional, and that there's a lot of things that kind of have to get broken along the way, and a plant would have to restore all of those things to return to like a C3 state. CAM is very different, actually, because in every cell, these plants still have a fully operational C3 cycle. So all they have to do to, reverse their metabolic adaptation and sort of go back to what they were ancestrally is just kind of turn off the CAM, and their C3 cycle will still work. So it's interesting. I think there's real non-parallelism there between these two different states. And I think that's also makes it more difficult for us to reconstruct the evolution of CAM, because I think there is a greater potential for reversibility. And so when we don't have perfect sampling of like a large clade of CAM plants, and we think, okay, should we call all the cacti CAM plants, even though we only know for sure that, it's been documented in 20 of them, and there's like 1,000, should we just code them all as CAM? I don't know, right? It's a judgment call. Whereas in this world of C4, if you've got a big C4 clade, you're like, oh yeah, they're all C4. So that's an aside, but yes, I think there is differences in the reversibility of these things.
Great, thank you. Then I want to go back to a couple of the questions that came in on the first part of the talk. So one is a pretty broad question, and so this one I think, at least for me, would be pretty tough to answer, but it's, do you have a simple way to explain the difference between systematics, clades, phylogenetics, and taxonomy? So that's a tough one, but maybe you can give us at least some initial thoughts on these different words that we use to describe what we're doing, I guess.
Wow. So the way that I'm currently thinking about it, and its part is only because I had to put this talk together, is I feel like systematic biology is almost like the umbrella under which a lot of these other things can sit. It feels the most sort of inclusive to me, and within that umbrella, you have people who focus really on taxonomy, which is really kind of the classification of organisms, right? And then you have phylogenetic biologists who are really interested in methods development and inference, and a lot of the stuff that we need to sort of answer a lot of our evolutionary questions. And you also have, in phylogenetic biology, I tend to view that as also, I don't know, applied phylogenetics, like ways that you can use phylogenetic thinking in other disciplines. That kind of falls into that category for me. What was the fourth one? There was another one.
It was clades, but
clades. Yeah. And so then what is this model clade thing? Is it a recapitulation of systematics, or should systematics be the big umbrella? Now I'm rethinking this, and the model clade approach is another element within that umbrella, which is more of like the Simpson description of what systematics is, about this gathering together and integrating knowledge. Maybe that's a way. I mean, this is something we should all work out. I don't exactly know, but it's really fun. When we were at SSB, when we were trying to come up with when our awards director was trying to come up with a rubric for the Ernst Mayr symposium, we're like, well, what's a systematics paper? And we had to like really just kind of sit down and try to figure out what we think systematics papers are. So yeah.
Yeah, great. I think that's a really great perspective. Okay, one more then, and again, this is related to the earlier part of the talk. Do you believe that a systematic view of the organism gives us a better biological perspective for the interpretation of comparative phylogenetic models?
Yes, of course. Yes, I do. Yes, hands down. And I am not a methods developer, I'm a methods user, right? But I do know that I think people who work very closely with organisms can look at the models of character evolution that we have to work with and feel like they could be improved. And I think a dialogue between people who are really buried in organismal detail and people who are developing methods is going to help us a lot. And I think that happens to some degree. I don't want to say that there's like this huge schism or anything, but I think that that kind of dialogue should be encouraged.
Great. Well, thank you so much, Erika. This was a really wonderful talk and a great perspective. And I thank you for taking time to talk with us about this. And thanks for all your work, and especially, interacting with me as past president and guidance of the society through a really crazy set of years now as we're into year 2 of this. So thank you so much for everything. Thank you for joining us today and giving this talk. And I want to remind everybody that the SSB business meeting will start in about 1 minute. So you can transition over to that Zoom link. The information was in the chat. Tracy put it in the chat. And so you can transition over to that Zoom link. And thank you all very much again.
Thank you. Thanks for coming and listening, and join our society. So fun. Okay.